Thank you very much. Thank you, Professor Dibo. Thank you, Ulrike, and thank you all who I already spoke to. It was great to be here, and it is still. I'm looking forward to learn more about your center after my talk by meeting with more people. Yeah, it's unusual times, but it's better than nothing. I look forward to meet many of you in person when opportunity arises, but this is great because I heard a lot about the university. Of course, I met a few people in person, and this was with opportunities for me to learn much more. Really happy about it. Yeah, so today we'll talk about our work in Stony Brook and Brookhaven on solving problems in catalysis with the help of machine learning. And I'll give an overview about our general interest in functional nanomaterials and linking structure and function, and the role that local structure plays in this problem, and challenges in finding out what role does it play. My general interest really are in solving problems, and so with this emphasis on solving structural problems, we started to use X-ray spectroscopy back in '95, a long time ago, where it became interesting to know what is the structure of mono and bimetallic nanoparticles, and we started to use XFs more and more actively. And it was the time when the analysis codes such as FEF just became efficient to analyze the structures beyond the first shell. And so it was a great opportunity to apply this new developments to learn about multiple scatterings and get access to coordination numbers beyond the first shell. And that opened opportunities because in catalysis, especially in nanoparticle catalysis, there are descriptors of catalytic activity that go beyond the general, very qualitative terms, such as whether it's spherical or oblate, whether it's core shell or random. It is important to know descriptors beyond that and link to theoretical modeling. For example, the interaction with support, the strain, the electronic properties such as D-band center and others are clearly and directly linked to various catalytic mechanisms in multiple reactions. And they're all coming from the details of local structure in this catalyst. And these details are very difficult to understand if the catalysts are at the nanoscale, because you can't probe them precisely for this purpose with any kind of imaging or scattering methods, or at least not yet. And so this is a clear challenge. And this challenge is really very universal, not only for catalysts, but other functional nanomaterials, such as, for example, the semiconductor quantum dots that we work with. And they also have their own problems, their own functionalities, and their own descriptors, such as the size of the quantum dots or shape or dopant to dot ratio. And if you can unlock this descriptors and have control over them, you can really advance in understanding how to make this design, how to design this, for example, not lead, not cadmium-based quantum dots, make them more environmentally friendly and even more efficient. If you could make, for example, a p-type or n-type quantum dot, you can make a junction and a transistor. So this is all coming to the question, what's the structure and what's the mechanism? Filtration materials, that's another project we're working on. Using porous nanomaterials such as metal organic frameworks or polyoxide metal blades, or hybrid materials where we functionalize metal organic frameworks with nanoparticles and learn how to capture and decompose catalytically toxic chemicals such as chemical warfare agents. And this is also a question of what knobs we need to tune in the local structure, such as pore size, pH, protonation, and others. And so this is a very important problem, of course, to solve. Other group projects all revolve around this issue: structure, function, relationships, nanoactuators, materials for optical recording. And so in our group, we have, it all, of course, depends on the current funding, but currently we are mostly busy solving problems in catalysis, in this electromechanical devices, and filtration materials. So we have postdocs and students working on it, and I'll introduce my group a little later. So this is a general sort of research activity, structure property mechanism, with the structure being the focus, front and center of our research. Because in the field of nanomaterials, as I already said, structure is the weakest link in this chain. If we knew the structure, we could probably easily advance to understanding what knobs to tune, but we don't know the structure. So in our work, we use many techniques. We use multimodal characterization, as many groups in the world do, including, of course, the groups in TACO, and we use operando instrumentation, operando characterization as a very important way of linking activity and structure, but it's still not clear how to understand the mechanism from that. We know that there is a link, but what is the causal relationship? What's the cause and effect? I'll come back to that. But I think the most interesting and right now, I would say, promising development is our efforts in solving the structure. And so for that, we have applied machine learning methods, such as neural network, for example, artificial neural network, shown on the top and shown here. And the purpose of the neural network is to find the unknown link between the spectrum, the data, and the structural descriptors. For example, parameters defining the three-dimensional structure of nanoparticles. And I will talk about it in detail soon. So let me pose the question that I just sort of generally formulated, but now I'll try to be more systematic, is if you have a nanomaterial, functional nanomaterial, such as catalyst, and you want to understand the relationship between its structure and function, how to go about it. So there are some attributes of this active structure, such as active species and active sites, and we need to know what they are, and we need to know how they transform in reaction conditions, and how to link them to the measured activity data. So the fact that it's really an interesting question, especially in the nanomaterial, is because there's not one, not two, but maybe several things that may change at the same time while you activate the catalyst and when it undergoes or participates in reaction. For example, in the top you see a collection of frames from electron microscopy experiments, in situ experiments, where carbon monoxide was flown at platinum nanoparticle on Syria. And you see that the image is so blurry, you don't make out of any positions of any atoms there. But once you add water to make it water gas shift reaction, you start seeing the fringes, you start seeing atomic positions. And the only change was that you added water to the reaction mixture. And so in this case, we understand after solving this problem, the structure mechanism relationship, that there are some sites on the particle that are active for forming hydrogens and carbon dioxide. And those sites are on the perimeter. You don't see them because they're really dynamic. You see that there are other other sites, but those on the perimeter where my cursor is, those are really going in and out of focus. And so this was pointed out to be the active sites. And there is an interesting mechanism there, and it was just published a few months ago, and my group member Yuan Li was the lead author on that. And so this represents to me a general sort of formulated research project. Research purpose is, if you have structural measurement and if you have activity measurement, how do you link them together to understand catalytic mechanisms? By the way, the reason that you don't see the atomic position so clearly on the CO is because of strong CO binding to the surface of the platinum nanoparticle. The repulsive interaction of CO molecule causes platinum platinum atoms move out of their regular positions, causing significant strain. That is the reason why you don't see anything there. But under the reaction conditions, CO spills over, forming converting to CO2 at the interface. So there is really no such disorder in influence under reaction conditions. That's why you see much more ordered images. So So here's a question: how do we obtain this information from the data? Because obviously we cannot really tell anything from these blurry images other than the fact that in one condition it's more dark than the fact that in the other condition it's not. What's the mechanism of that? So, so the question is primarily, how precisely can we detect the structure from this material, in this material? And because the imaging is not so efficient for that, it's not so effective, we need to go around that that problem and find an alternative. And so the alternative is, let's rely on the spectrum as the source of information for the structure and activity. Because if you measure the spectrum, such as X-ray absorption spectrum, in operando experiment, you can measure both of them in the same condition. So that sounds promising, but the spectrum and the structure contain different information. The actual material has atoms, and each atom has X, Y, and Z coordinate. And so if you have a particle consisting of, let's say, 40, 50, 100 atoms, you have a lot of information to stored in it. But in the spectrum, you can't expect that it will give you X, Y, Z coordinates of all atoms. So you need to really understand what information is encoded in the spectrum. That's number one. Number two, suppose you know the answer about the structural descriptors. What do you do with that information? In order to understand the mechanism, it's not enough to get the structure-activity relationship. You need to explain it. So you need to find a causal relationship. Why, in what order, what are the sequence of elementary steps, how does the reaction progress? So that means that you need to find the key descriptors, the key factors that are responsible for the activity. And that's a question to theory. What are they? It can be coordination numbers, it can be some generalized quantities, it can be interatomic distances, or all of the above. So I know it's very abstract, but I'm trying to use specific examples from the published data. But in the second part of my talk, I'll actually walk you through the recent experiment, recent research, in which we actually from the beginning posed this question: how to make a framework of research for catalysis, for a catalysis problem that we can solve using this multimodal machine learning assisted method. So coming back to this paradigm, if you want to find those descriptors, you can't really get them from the structure because you don't know what to look for. There's so much information encoded there. But again, if you use the spectrum as a source of information, it has much fewer quantities encoded in it. And if you can map them on the space of those descriptors, then you can close the loop. So here we have this general framework. Instead of going for the structure, let's go around it. Let's measure a spectrum. Let's use descriptors encoded in the spectrum as the source of theoretical modeling. And the theory will explain catalytic activity, which will be measured. So by going around the main question here, what is the structure? We will be able to answer the more important question: what's the explanation for the reaction, for the mechanism? Here is the problem. Theory can very well work in this space because theory can start with certain model and calculate using DFT or microkinetic modeling reaction pathways. But how does theory know what is the realistic model of the structure and what are the descriptors of the structure? That is the missing link here. And we're solving this link by using machine learning. So I'm gonna show how we do that. As I already outlined the main idea that we link the spectrum to the structure using the neural network, but I should give credit to the group members who were in the beginning of that. And Yanis Timoshenko was first who understood the concept of neural network in application to structural characterization. That was the product of few months of brainstorming together with data scientists from Brook and our group, where we got together once a week in a closed room and like educating each other first about what is X-ray spectrum, what is structure, what is neural networking. And people who worked with us from the other part, from the neural network part, they were educated as Google data scientists, and so they primarily applied it to like natural language processing and problems related to like analysis of basically data mining, bioinformatics. But we learned how to apply it to our problems. So the idea is that you create a function that takes input, which is the spectrum, and the output will give you the information about the structural parameters. And I'll show how it works in a moment. But before that, I wanted to show to you that this approach is quite universal. And once we understood how to apply it to nanoparticles, we expanded it to other nanostructure systems that have the same potential. For example, from monometallic, we went to bimetallic nanoparticles and learned about their restructuring by extracting coordination numbers directly from the Zanes. And in doing so, we were able to find an alternative to XOFs, which is the more traditional method. But in using X-ray absorption fine structure, we're limited to very well-defined data, very high-quality data that usually can be obtained only in very friendly conditions, concentrated samples, low temperature, and very uniform samples. And so when you do experiments in operando conditions, you usually have something just the opposite. You have high temperature, low loading, and harsh reaction conditions in general, including reactor walls and some limitations in geometry. And so we don't get that luxury of getting good quality XOF spectra to obtain coordination numbers and distances and other important parameters. And so we resorted to look into Zanes as an alternative, and it was very rewarding. For once, Zanes is not limited to just concentrated samples, because even in the low concentrated samples, you can see the edge, you can see sharp features, and that's all we need to make it work in those examples that we tried. And also, of course, just like XOFs, we can do it in various realistic conditions. So after obtaining this method, after developing this method, we started to apply it to different catalytic systems, from monometallic and bimetallic nanoparticles. We extended it to size-selective clusters. We applied it to some materials in which XOFs would not even work, such as disordered, strongly changing materials, such as during phase transition, where XOFs would not even be useful. And lately, we had the first success in using it for single atom catalysts. I didn't even have time to add the example because the paper is still in review, but I can tell you more if we have a chance to discuss it during the next meeting I have with this group. So the other question we ask is, okay, we have this well-defined algorithm that you have an input and you map it on a certain output. But how do we know what to map it on? And how do we know how much opportunities we have to map it on, let's say, more things? Not only coordination numbers, but maybe interatomic distances, maybe electronic properties, maybe some other intricate details of nanoparticles. So for that, we need to know how much information is in the spectrum. And we used a different approach completely, which is called unsupervised or semi-unsupervised machine learning based on the autoencoders. The autoencoders link the data to the data. So for example, if you have a database of faces in the some criminal database like FBI database, they don't encode, they don't record all the pixels. They make it compressed so that only a small amount of information will be sufficient to recognize, for example, if a new face appears, does it resemble any entry in that database? So it's based on the ability of neural network, machine learning, to recognize how many key variables are required to reliably reproduce the entire data. It's very similar to what we learned from, for example, principal component analysis, which I'm sure many of you are familiar with, is that if you have a sequence of spectra, let's say thousands of spectra, how many unique species are there during the entire transformation? And using principal component analysis, you can tell that, for example, the 5 or 6 eigenvalues are much more important than others. And you can tell that the dimensionality of your problem is really like 5 species, not thousands. So that's a similar approach kind of in spirit, but it's non-linear. That's why we use autoencoder as opposed to PCA. So this is the kind of big picture summary. And since 2017, we have even a patent on the method and a few papers on different applications. So why to go back from the spectrum as opposed to going forward from the structure? It's really a philosophical question. I like this story from Gulliver's Travel, where there were groups divided in how they break the egg. You all remember that? The ones from the ones that broke the egg from the big end or from little end, and they fought each other. And in many scientific communities, not only in XAF's community, there was not a fight, of course, but of course discourses like fitting in K space is better or fitting in R space is better, how to model Zanes from the structure to spectrum or vice versa. And the latest part is what makes it most promising, I think, in this space. Because if you start from the structure, it's very subjective approach. What if your structure is wrong? And what do you know about the quality of the agreement between the model and the experiment? It's never going to be as perfect as the fitting of XAF's data, for example, because in XAFs you always strive for a good fit. But here in Zanes, using any theoretical code, you will never get a good fit. So what's the criterion of agreement between model and experiment? So in the case of modeling of Zanes, this type of paradigm between big end or little end is exactly what I just described. Is, do you start from the model and obtain the spectrum? You can. There are many codes that can do that, but it's not a unique approach. You don't solve uniqueness. You don't guarantee uniqueness. And maybe there is another structure which also matches with the data. And what do you do about not knowing that? And this is the structure to spectrum kind of approach. And the other approach is what we developed. We map the spectrum to the structure. And we know that we cannot do it in any kind of fitting setting, because there is no analytical equation that we can fit to the spectrum and obtain the structure. Anybody who analyzed XAFs knows that you have XAFs equation. You introduce fitting variables, you run it through the fitting program, which are many out there, and you get the best fit parameters. And we cannot do it for Zanes. So how do you invert? And that's why we found this machine learning is really a good match for this problem. And the way it works is this. In general, a neural network, many of you probably are familiar with that, is the function that maps or calculates output by knowing the input, except instead of using some kind of analytical or implicit relationship between input and an output, it's done by a very simple sort of sequence of transformations, where there is a prescription how to obtain the next value in the next layer, and each circle here is a node which has a number. And so how to obtain another number, it's done by using a function of all the nodes, adding together some kind of linear combination using weights, and the result will be a new value in this second layer, first node. And then the same for the second node with different weights and another node. And after that, the values in the output will be obtained. So what it does, it gives you a lot of flexibility, much more than any kind of analytical function, that you can in principle map anything on anything. Obviously, there should be some hidden relationship between that anything and anything. Otherwise, you will not ever never converge. Like you can't map spectrum on the brand of coffee I just drank because there is no correlation. But you can map a spectrum on the coordination numbers. Why not? Because it informs the spectrum. And so the program, the only thing that the program needs is training. It needs to have some kind of algorithm or criterion to know, after you initialize those weights and biases, how to adjust them to improve the predictability. And this is done by using one of the many conventional optimization methods. In this case, it's called backpropagation, where there is a cost function or penalty for having a wrong parameter or wrong hyperparameter in this neural network if your disagreement between the predicted value and actual value is big. And so for that, you need to have labeled data sets. If you don't have labeled data sets, you can't use artificial neural networks. So the labeled data sets consists of spectra for which we know what the output is. And by feeding to the neural network many such spectra, the agreement between the prediction and the label gets better and better. And finally, the neural network is stabilized and gets you this fixed parameters of each node, not each node, but each parameter corresponding to this transformation shown by arrow, how to obtain one value from the other value. And then it's ready for predicting on the unknown spectrum. So this was the method. We applied it first to predict the three-dimensional structure of metal nanoparticles. We had many spectra of platinum nanoparticles calculated theoretically. And there is a separate sort of know-how that we developed to make sure that the spectra are unique and different. Otherwise, if they're all are similar to each other in some features, then there will be no learning by feeding more spectra like that. And actually, it's a limitation, because in the nanoparticles, what can you do to make them different? You can make them bigger, or you can make them flatter, or you can make the changes in their shape. And if they're small, there will be some contrast in the average spectrum as a result. And you can use it to train neural network. But if they get bigger and bigger, the contrast gets smaller and smaller because the surface to volume ratio gets smaller. So we quickly understand that this is a limitation for theoretical modeling, because you can really run out of possible training sets very quickly. So we developed a combinatorial approach. What it does is that we have several nanoparticles, let's say 30 or 50 of them, which have different shapes. And for each nanoparticle, we could identify an atom. For example, where my pointer is, it's a corner atom. And the spectrum for this corner atom is distinctly different than others. Like this is the one that I'm showing with the pointer. And the spectrum for the atom here on the facet, triangular facet, for example, green, perhaps it's this one, also very different. So by using a combinatorial approach, we mixed and matched spectra from different sites on this generated nanoparticles, and we could generate hundreds of thousands of training sets. So by that, we were able to train our neural network. And then when we fed our experimental data, it was able to predict the same coordination numbers as we could obtain independently. And we published it over many papers in well-defined nanoparticles that back then we did not know that we need to reanalyze them, but now we analyze them by Zanes and obtain the same results. So that's how we knew that the method works. So this is the first part of my story. I'm moving now to the, probably the main part is how do we decode reactive structures using this approach? I'm sure that people may have questions about it, and so probably the option would be to put them in the chat, right? But let me then continue and answer all of them together if you don't mind. So this is sort of a tool, right? But it can quickly get sort of less exciting after you publish one paper and then the other paper, and then it becomes really boring, right? Because yet another paper of how to invert a spectrum, what is so special about it? So we really stopped quite soon applying this to other systems because we realized it's going to be not really novel anymore. And we decided to use it as a tool to solve real catalysis problems because that was the missing link in solving the structure-function relationship, how to understand the relationship between the spectrum and the structure. So I'm going to show to you the very recent work that was just accepted to Nature Communications, done by a very large collaboration. It's a collaboration as part of the Energy Frontier Research Center at Harvard, centered at Harvard, and I'm part of it, where we have a 4-year program. It's already the end of the second term, so it's already 8th year. And a year ago, the director, Cynthia Friend, decided that we will have seed projects to discuss or to think about some future directions. And so I was leading the seed project on what is called decoding reactive structure, which is the subject of my talk, how to generally solve, it's kind of like high aspiration, but it is what we really try to do, how to solve this general structure-function relationship using this machine learning approach as multimodal characterization. And that's what I'm going to talk now about. And our center deals with dilute alloy catalysts, and this is the theme. And they are successful in many ways to tune activity and selectivity of catalysts. And the biggest part of it is due to this balance that dilute alloys have. When you have, for example, a majority component, like for example, silver, 111, will have very high activation barrier for hydrogen dissociation. And it would be much lower. And sorry, let me go to another extreme. If you have palladium, not silver, it will have almost no barrier, but very strong binding of hydrogen atoms to palladium surface. So it's not good either. And if you have palladium dilute on silver surface or on gold surface, it will be just right. It will have small barrier and small binding energy. So you can really use it for reaction. And that was shown by Randall Meyer and his co-authors 6 years ago, 7 years ago. So in our work in IMASC a couple of years ago, we found strong dynamic effects corresponding to this palladium, dilute palladium alloys. In this case, we experimented with palladium and gold. At different temperatures, in different reaction environment, palladium ions, palladium atoms redistributed on the surface of gold. And we did it using nanoparticles, not surfaces. And so, for example, under hydrogen, the catalyst was not active, and this was the example, I think it was hexyne hydrogenation reaction. And under oxygen, it became active at high temperature and partially active under CO at low temperature. So there was clear effect of some kind of restructuring of the alloy components on the reactivity. And we wanted to understand also how to capture that and how to understand the role of that for catalysis. So in our seed project, in the project that I told you we started about 1.5 years ago in IMASC, my postdoc Anna Plonka and the student Nick, they use this reactor, very typical workhorse reactor, we would call it, called Clausen cell also, where we do operando experiments using X-ray spectroscopy, also X-ray diffraction, and measure activity in the same time, simultaneously. So this reactor was used to measure activity on for hydrogen dissociation. It was actually not hydrogen dissociation, it was HD exchange, because it's very difficult to detect hydrogen dissociation products by mass spec. But if you have hydrogen and deuterium, of course, detecting HD is easy. And so the questions were, what are the active species? And if we had this dilute palladium and gold, there could be many possible answers, or it could be a combination of the answers. It could be monomers, dimers, trimers, it could be bulk palladium, it could be something that is a combination of that, or it could be just gold. And what are the reaction mechanisms there? And so the challenges are, of course, that not only it's a dilute catalyst, we don't have a very clear way to detect the position of palladium on gold, and we knew that the answer depends on the knowledge of that. We also know that it may restructure in reaction conditions. It may restructure in the pretreatment process, and I already showed to you the evidence that it does. And therefore, it's a very dynamic system, and it's very difficult to analyze it by conventional methods, especially if you don't know the structure. So our approach was that let's link, just like I showed to you in that sort of schematic, let's link structure, activity, and theory together. And in order to understand the structure, we will use this machine learning assisted ZANEs, because there is no other resource of structural information. You can't get structure of dilute palladium and gold by any other tool in reaction conditions. So the activity measurement was studied by mass spec, and you see here the example of HD formation at different temperatures. It was clearly some kind of deactivation going on, because in the initial stage, after activation in oxygen and preliminary annealing, so we have 1, 2, 3, 4 different states in which the catalyst was measured after different treatments. So the first treatment was in oxygen, and the sample was called S1. The second treatment under hydrogen, S2, and then repeated treatment under hydrogen, S3. And so after oxygen treatment, the catalyst had very strong change because initially it had very high activation energy and then it dropped, and you can see it from the data. And so the activity and kinetic parameters decreased on hydrogen treatment, and we wanted to understand partially why, not only what the reaction mechanism is, but what is the role of structure of palladium for this deactivation. The second set of data, the same sample was brought, the same samples, the same reactor was brought to the beamline. We collected that NSLS2 under in situ conditions. We obtained data both in XANES and XAFs. And you see that there are clear changes, and XAFs is a more traditional technique. So we can start with that and show to you that in the process of progressive hydrogenation or exposure of the sample to hydrogen before the reaction, the peaks corresponding to the first nearest neighbors, in this case it's primarily palladium gold because palladium was very dilute, increased. And that would be consistent with palladium having more neighbors. That's sort of intuitive. And not only more neighbors, more gold neighbors, because you would not see palladium bonds in 8% palladium, very dilute. So that's qualitatively consistent with the explanation for restructuring, but we wanted to have it quantitative. And so we did full XAFs fitting, and we found that the uncertainties and the best fit parameters quite large, and so the results really overlap with the uncertainties, and it's not nice. But when we did this, when we performed this machine learning analysis of the XANES data, which is shown on the top left, which clearly show very interesting and very easy to understand trend, then we obtained much better and much more systematic answers. And they were consistent with XAFs, but with much smaller uncertainties. So look here, the dashed curve corresponds to palladium foil. It's a reference. That's the white line, or the maximum of the edge of the X-ray absorption. And the blue one corresponds to the sample that was just oxidized. And it has, yeah, and it has the most proximity to, if I can say that, to palladium foil, which means that of all other spectra, it resembles palladium foil the most, which means that palladium there is likely to form palladium-palladium bonds or palladium ensembles. And it's all relative, of course, compared to what? Compared to other states, like such as S2 and S3. And in those states, the effect is less because it moves away from the direction toward palladium foil. So it becomes more dilute, more alloy. And so XANES analysis by using our neural network approach showed just that in the early stage of the process, palladium gold was lower coordination number than in the hydrogen treatment. On the hydrogen treatment, palladium dissolved better in gold than under oxygen treatment. So this is the meaning of this result. And in fact, you can even detect the change in palladium coordination number. When you have oxidation, palladium is segregated closer to the surface and has other palladium atoms nearby. That's why this coordination number is detectable. And then after hydrogen treatment, it dissolves and it's primarily surrounded by gold. So it is qualitative still, because we don't know what exactly happens, how many ensembles of which kind are there. And so we applied quantitative analysis of XANES. I already showed it to you earlier. Here in the center of the slide, here on the left, and in the center, it's basically a summary of our machine learning tests, showing that theoretical prediction and experimental trends are similar. So we can trust neural network to tell us those answers. But I don't want to go to too much details on that. This is already sort of optional. I cannot answer that if people ask questions about it. But also I wanted to give credit to another component of this research, which is we can't go blind about this kind of very detailed structural analysis, because the homogeneity or heterogeneity is very important to know, whether it is self-similar alloys, you know, from particle to particle, or there is very broad distribution of sizes, compositions, and whatnot. And if you don't know that, average information from XANES or XAFs is misleading or doesn't tell us anything. So fortunately, we had very good group here, a very good team in our project, and with access to excellent electron microscopy facilities. And as you see here, the distribution of gold and palladium was quite uniform. within the particle and also across the particles. I don't show it here, but the distribution showed that up to 90% or more were homogeneously distributed. And I want to give credit to Jessie van der Hoeven. I know that she was in this group with Professor Diebold as a master student, so it's a coincidence, but nice link to you. And Alexander Foucher from Eric Stock's group. And I'm moving now to the theoretical part. So after this information is known to us, what to do with that? So what we are ready now to consider is that theory does not start with some kind of subjective model that is not informed by anything. And even if it was informed by some surface science experiments, it's still far from the reality of real operando catalysts. But now it is informed by real operando catalysis studies. And what we know from them, from Zane's analysis particularly, that the oxygen treatment and hydrogen treatment have different structural, correspond to different structural distribution of palladium. We also know from activity measurements that activation energy of about, in this case in electron volts, about 0.65 or 0.7 electron volts under oxygen treatment regime, and about 0.35 on a hydrogen can be model theoretic. So when theory can start with just few models, it's much better than having arbitrary starting structure. And in this case, we had very limited possible ensembles to consider because this number was constrained by our X-ray measurements. And so, as you see, considering different processes of hydrogen or HD exchange, starting with the dissociation of hydrogen and deuterium, the recombination, all these energies were calculated and are shown here as the vertical bars. And they're calculated for different surfaces, 111 to 11331, as different colors. And so you see here that, for example, for a large surface of palladium, a monolayer, for example, we have very high effective or apparent activation energy, inconsistent with experiment, either for oxygen treatment or hydrogen treatment. But when you have a trimer, the activation energy predicted theoretically is just right. It's just in a good agreement with what was obtained under oxygen treatment. So this is consistent with formation of this type of trimers, which were this palladium ensembles on the surface. And we can say with certainty that trimers were dominating. It was not a mixture of trimers and dimers of some sort, because with dimers we would have much lower activation energy, and we would not be able to easily distinguish between dimers and monomers, because for both of them we would have similar activation energy consistent with experimental data. But it is still very informative conclusion from this, because we can pinpoint what ensembles are formed under which regime. And so by doing this, I hope I convinced you that we can really do this analysis in the framework shown here, where instead of knowing the structure by directly measuring it, which sometimes is not possible, we go around it and measure indirectly spectrum using machine learning to decipher it, and use the information as the input for theory that will model the experimental activity. And that will close the loop, and we will understand all we want to know about reaction mechanisms. In the part which is still sort of not quantitative, I wanted to show this analysis that my student Nick Marcella did, where we can link the experimentally measured coordination numbers by machine learning Zanes to possible structural motifs representative of these numbers. Of course, these coordination numbers are just, average information. So if you have some distribution, they may still end up to be the same, but therefore it's a representative, not exhaustive search. But at least it gives us some speciations that are typical in this material. For example, we can say how many monomers are there in different regimes. Literally, we can use the same particle size and shape as we can detect from electron microscopy. And we can place those monomers or dimers or trimers in just the right quantities as dictated from this Zane's analysis. And so this is a very detailed three-dimensional, maybe slightly oversold, because as I said, there's still some vague question how broad is the distribution of compositions. But as a representative figure, I think it can do the job. So with that, we can say that we obtained reactive structures. We say that they're responsive to the environment because they change dynamically in response to their environment. And in principle, we can plan different reactions depending on what catalytic species we put on the surface. And if for some reactions, monomers are more active than dimers, we know how to do that. And so we can use the same catalyst and tune it for different reactions. And we can monitor in reaction conditions what catalytic species are on the surface by using these techniques. So that's an added sort of maybe a very futuristic goal, but conceptually it's doable. People now talk a lot about autonomous experimentation, and that also is compatible with possibly doing it autonomously, because if, for example, the catalyst deactivates and we want to know why, we can sort of have a feedback between the spectral analysis that tells us why it deactivates and change something with the reaction mixture or reaction conditions to reactivate, because we can now understand why it deactivates. So the conclusions of this, and I'll have time for questions, are that our reactive structures are obtained and they satisfy both structural and kinetic. I didn't say anything about kinetic modeling to save time, but we've done microkinetic modeling of full reaction pathways using the DFT calculated barriers. And they all confirm the measured activities, which was much more accurate way of modeling it than by using DFT, but that's in the paper. And so we can also say that neural network Zanes is not just some kind of scientific curiosity, but it's really a useful tool to link structure and activity studies. And also it can measure coordination numbers quite accurately and with lower uncertainties than by XSubs. We can tune, and we showed that we actually change the surface ensembles by using different pretreatments. By the way, what I didn't show here, but it's in the paper, is that with 8% palladium, which was the concentration studied here, we were able to show that initial surface composition was due to palladium trimers. And we know that palladium trimers have much higher activation energy for hydrogen dissociation than dimers and monomers. And so as additional sort of reality check, we had another experiment done with 4% palladium. And 4% just doesn't have enough palladium to form those trimers. And the activation energy was much lower, was somewhat halfway toward the deactivate well to the monomer one. So that was another proof of sanity check, that it is indeed the right model, that under oxidizing conditions, there are trimers on the surface. And so that's the main last conclusion. And I wanted to summarize by showing who are the collaborators here. It's a big part of IMASC Center. The PIs are Joanna Eisenberg, Jürgen Binner, Anibal Voskoboynik. They are all PIs of IMASC. Not all of them were involved in this work. In this work, Joanna, Cynthia Fran, Boris Kozinski, Philippe Sauté, Eric Stock, and Jason Weaver, and their students. And I also want to give credit to Jesse, who did a lot of work on the system even before we started. And my group on the right includes research scientist Yuan Li and several postdocs and graduate students, all heavily involved in this type of activities. So I also want to give credit to Beamline and the synchrotron catalysis consortium that we built for solving this kind of operando problems. And I would like to also thank you for your attention and the opportunity to present this work. Thank you very much.